Good morning, everyone. Welcome to today's presentation. I'm delighted to have you all here. Today, we will be discussing the qualification of pH meters, an essential topic in analytical chemistry. The focus will be on understanding how pH meters are calibrated and maintained for accurate measurements. We'll cover the basic principles, the proper procedures, and some best practices. So, let's get started and explore this important subject together. Today, I'll be discussing the scope of our qualification process for laboratory pH meters used in quality control analysis. The main goal here is to ensure that the pH meters we rely on are accurate and reliable. This involves verifying their accuracy, precision, linearity, and overall performance. By doing this, we can confirm that the measurements they provide are trustworthy. Our objective is not just about testing the equipment. It's also about making sure we stay compliant with pharmacopoeial standards and good manufacturing practices, or GMP. These are critical to maintaining the integrity of our quality systems. Now, why is pH measurement so important? Well, in pharmaceutical quality control, pH is a key analytical parameter. We use it to test raw materials, monitor in-process samples, evaluate finished products, and even check water systems. Getting accurate pH readings is essential because it impacts product quality, stability, and ultimately, patient safety. If our pH measurements are off, it could lead to incorrect conclusions about the quality or stability of our products. So, maintaining rigorous qualification processes helps us ensure that every measurement we take contributes to producing safe, effective medicines. Let's take a look at some of the key applicable guidelines that govern our work. First, there's USP 791, which focuses on pH measurement. This is crucial because pH affects the stability and efficacy of our products. Then we have the European Pharmacopoeia, specifically EP 223, which provides standards for pH determination across European markets. Moving on, the ICH-Q2, R1, guideline is all about the validation of analytical procedures, ensuring our methods are reliable and reproducible. Additionally, the EU GMP Annex 15 outlines the requirements for qualification and validation of facilities and processes, reinforcing quality assurance. Lastly, the FDA 21 CFR Part 211 sets the regulations for good manufacturing practices in the United States, covering everything from raw materials to finished products. Connecting all these, it's clear that compliance with these guidelines is essential for ensuring product quality, safety, and efficacy across different regions. They form the backbone of our validation and quality assurance processes, aligning with our overall commitment to excellence in pharmaceutical manufacturing. Let's take a closer look at the principle of pH measurement. So, a pH meter essentially measures hydrogen ion activity in a solution. It does this using two main components: a glass electrode, which acts as the measuring electrode, and a reference electrode. The glass electrode is sensitive to hydrogen ions, while the reference electrode provides a stable baseline. When these electrodes are immersed in a solution, they generate a potential difference, or voltage, that corresponds to the pH level. This potential difference is then converted into a pH value using the Nernst equation, which relates voltage to ion concentration. Now, an important factor to keep in mind is temperature. Temperature changes can affect the electrode response, so temperature compensation is critical to ensure accurate and reliable pH readings. Overall, understanding these core principles helps us appreciate how pH meters provide precise measurements essential for many applications. Today, I want to walk you through the qualification life cycle, which is a crucial part of ensuring our systems and equipment are reliable and compliant. The process is divided into three main stages: installation qualification, or IQ, operational qualification, or OQ, and performance qualification, or PQ. First, with IQ, we focus on verifying that the equipment or system has been installed correctly according to the manufacturer's specifications and regulatory requirements. It's about making sure everything is in the right place, properly calibrated, and ready to operate. Next comes OQ, where we test that the equipment functions as intended under normal operating conditions. Here, we validate the critical parameters and controls, ensuring the system performs consistently and safely. Finally, PQ is about confirming that the system consistently produces the desired results in real-world conditions. It's the stage where we demonstrate that the entire process, under typical production scenarios, meets quality standards and specifications. Connecting this to our overall theme, these qualification stages form a continuous cycle of validation and verification, helping us maintain quality, compliance, and confidence in our processes. Each stage builds on the previous one, ensuring that from installation to actual performance, everything functions seamlessly and reliably. Let's talk about installation qualification, or IQ. This is a crucial step to ensure that our instruments are set up correctly from the start. First, we need to verify that the instrument has been installed exactly as outlined in the manufacturer's manual. This helps us confirm that everything is in the right place and properly assembled. Next, we check that the model, serial number, and software version match what was specified or ordered. This way, we avoid any mix-ups with equipment or outdated software. We also verify the power supply and environmental conditions around the instrument. Proper power and suitable environmental factors, like temperature and humidity, are essential for accurate and reliable operation. Calibration buffers used during setup should be traceable to certified standards, ensuring their accuracy and integrity. Lastly, we confirm that all necessary standard operating procedures, manuals, and calibration logs are available and up to date. When all these checks are completed, the IQ is considered acceptable. Remember, documenting and getting approval for these installation checks is vital. It's not just about setting things up, it's about creating a solid foundation for quality and compliance throughout the project. This step ties directly into our overall goal of ensuring reliable, validated instrumentation that supports our quality standards every step of the way. Now, let's talk about operational qualification, or OQ, which is a critical step in ensuring our analytical systems are functioning correctly. When we perform OQ, we focus on several key parameters. First, calibration accuracy. This is about making sure our instruments are providing precise measurements. It's essential because accurate calibration underpins the reliability of all our results. Next, we verify the electrode slope, which confirms that our electrodes respond consistently and linearly to changes in analyte concentrations. This helps us catch any issues with electrode performance early on. We also check the offset, or asymmetry potential, to identify any baseline shifts that could skew our measurements. Ensuring this is within acceptable limits helps maintain data integrity. Lastly, temperature compensation verification is vital because temperature can influence our readings. By confirming that our temperature adjustments are working properly, we ensure our results remain accurate regardless of environmental changes. All these parameters are interconnected. They collectively assure that our equipment is performing optimally before we proceed with sample analysis. Remember, thorough OQ is the foundation for reliable, consistent data in any analytical process. Let's now go through the calibration procedure, which is a crucial step for accurate pH measurements. As per our SOP, you can perform a 5-point calibration if needed, but typically, a 3-point calibration is sufficient. The key here is to use calibration buffers that represent the pH range you're working with. Usually, we use pH 4.01, pH 7, and either pH 9.18 or 10.01. These buffers help ensure your electrode is properly calibrated across the spectrum. Now, the procedure itself starts with rinsing your electrode thoroughly with purified water to remove any contaminants. Then, you'll immerse the electrode in the pH 7 buffer. This is your neutral point, and adjust your meter accordingly. After that, you'll move to the acidic buffer, pH 4.01, and then to the alkaline buffer, either pH 9.18 or 10.01, calibrating each time to get accurate readings. Remember, it's important to record the stabilized pH values at each step to ensure your calibration is reliable. This process helps us maintain measurement accuracy and consistency, which is vital for the integrity of our results. Proper calibration connects directly to our overall goal of obtaining precise and reproducible data, so taking the time to do it correctly is always worth it. Let's now turn our attention to calibration accuracy. This is a critical aspect because it directly affects the reliability of our measurements. When we talk about calibration accuracy, we're referring to how closely our measurement system aligns with a known standard or true value. Essentially, it's about ensuring our instruments are giving us correct and consistent readings. High calibration accuracy means we can trust the data we're collecting, which is vital for making informed decisions and maintaining quality control. Throughout this presentation, we've emphasized the importance of precision and consistency, and calibration accuracy ties directly into that. If our calibration isn't accurate, even the best sensors can lead us astray, affecting the overall performance of our system. That's why regular calibration checks and adjustments are essential. They help us maintain that high level of accuracy over time. So, when we think about calibration accuracy, think of it as the foundation that supports the integrity of all our measurement processes. Now, let's move on to slide 10, which focuses on electrode slope calculation, specifically for OQ, or operational qualification. This step is crucial because it helps us understand how the electrode behaves under certain conditions, ensuring our system performs reliably. When we talk about the slope, we're essentially looking at how the electrode's response changes with variations in the measured parameter, usually pH or other ion concentrations. Calculating this slope accurately allows us to verify that the electrode is functioning within the acceptable range, which is vital for maintaining quality control. It's important to remember that a proper slope indicates the electrode's sensitivity and proper calibration, which ties directly into the overall quality assurance process. So, in essence, this calculation is about confirming that the electrode's response is consistent and predictable, supporting the integrity of our measurements. By ensuring we accurately determine the electrode slope during OQ, we can confidently rely on our measurements for critical decisions. Now, let's take a closer look at performance qualification, or PQ. This is a critical part of validating our analytical methods to ensure they perform reliably in routine use. When we talk about PQ tests, there are a few key components to keep in mind. First, we have repeatability, which essentially measures the precision of our method when we perform multiple tests under the same conditions. This tells us how consistent our results are. Next, there's intermediate precision, which takes it a step further by assessing the method's performance across different days or with different analysts. This helps us understand if the method remains robust over time and across personnel. Finally, we have routine sample simulation, where we simulate real-world sample testing to confirm that our method can handle typical samples in everyday scenarios. All these tests are designed to confirm that our method isn't just accurate in ideal conditions, but also dependable and reproducible during regular operation. This consistency is fundamental to maintaining quality and compliance in our work. So, in essence, PQ ensures that our analytical methods are not only validated initially, but continue to perform reliably over time, supporting the overall integrity of our quality system. Let's take a look at this example of precision, specifically repeatability. When we talk about repeatability, we're referring to how consistently a process or measurement can produce the same results under the same conditions. It's a key aspect of precision because it shows that our method is reliable and stable over time. Think of it as hitting the same target multiple times. The closer those shots are to each other, the higher the repeatability. This consistency is crucial for quality control and ensuring that our products or processes meet the same standards every time. By emphasizing repeatability, we're reinforcing the overall theme of precision, highlighting that accuracy isn't just about hitting the target once, but about consistently achieving the same results. This reliability builds confidence in our processes and helps us identify areas where we can improve stability and performance. Let's now talk about routine checks and requalification, which are vital for maintaining the accuracy and reliability of our measurements. First, daily calibration before use is essential. It ensures that our electrodes are providing correct readings each day. Using fresh, certified buffers is equally important because it guarantees that our calibration standards are accurate and traceable. Monitoring electrode performance regularly helps us identify any drift or degradation over time, so we can address issues early before they impact our results. Requalification is a critical step that we perform after certain events. For instance, if an electrode has been repaired or replaced, requalification confirms that everything is functioning properly again. It's also necessary annually, or as specified in our SOPs, to ensure ongoing accuracy over time. And of course, after any out-of-specification results or deviations, requalification helps us verify that the system is back within acceptable parameters. All these checks and requalification steps are part of our overall quality assurance process, reinforcing the importance of consistent, reliable measurements in our operations. In conclusion, I want to highlight that our pH meter has been qualified according to USB and GMP standards. This means it has met all the necessary requirements to ensure it's reliable and accurate for pH measurement. Such qualification is crucial because it supports regulatory compliance and upholds data integrity, which are essential in our industry. Now, moving on to documentation and final thoughts, there are several key documents we need to keep in place. These include IQ, OQ, and PQ protocols and reports, which demonstrate that the equipment has been properly installed, operated, and performed as intended. Calibration and slope records are also vital. They ensure that the pH meter remains accurate over time. Additionally, buffer certificates of analysis and traceability records help verify the standards used and trace back measurements if needed. Lastly, maintaining logbooks and standard operating procedures is fundamental for consistent operation and compliance. Together, these documents and processes create a solid foundation, confirming that our measurement system is robust, compliant, and reliable, integral to our overall quality assurance efforts. For further support, please don't hesitate to reach out. If you have any questions or need additional information, I'm here to help. Remember, our goal is to ensure you feel confident and well-equipped moving forward. Feel free to contact us whenever you need assistance. Thank you so much for your time and attention today. I truly appreciate your engagement and look forward to supporting you in any way I can.